To begin, we will discuss the background of antifreeze protein. As competition for space and resources increases, animals need to adapt to fulfill different niches. A paper written by Sudaria and Sunkasula stated that the antifreeze proteins have been found in bacteria, algae, fungi, plants, insects, and polar fishes, but they were originally found in Antarctic marine fish. This allows them to occupy and survive in an extremely cold environment. The notothenoid is a suborder of fish that are native to the Southern Ocean, mainly in the Antarctic region. Within the suborder, there are 8 families, 3 non-Antarctic and 5 Antarctic. The family that we will be focusing on is the Notothenidae, which is one of the 5 families that are native to the Antarctic. Due to the variety of organisms that have antifreeze proteins, there are multiple categories: AFP1, AFP2, AFP3, AFP4, hyperactive AFPs, and anti-freezing glycoprotein AFPs. The main difference between all of them is the length of the protein. The hyperactive AFPs are the longest at 32 kilodaltons, and AFP1s are the shortest at 3.3 to 4.5 kilodaltons. More specifically, the Notothenidae have antifreeze glycoproteins, or AFGPs. These proteins work by ice crystals entering the bodies of the fish, and the AFGPs bind to them via an absorption inhibition mechanism, which inhibits further growth of the ice. It is the absorption that inhibits the thermodynamically favored growth of the ice crystals. The antifreeze proteins allow for cryopreservation via thermal hysteresis as well. This is the separation between the temperature at which a liquid is melting or freezing. In 1986, De Vries used nanoliter osmometry to study the shape of ice crystals. Now, this method is a commonly used technique in the research of antifreeze proteins. In this process, a temperature-controlled chamber is used to hold a sample. Then the chamber slowly cools, and the melting point is determined when the ice crystal is fully melted. As written by Buckley and Lilford, because of this process, researchers were able to discover how antifreeze proteins can depress the freezing point of a solution while maintaining an equilibrium melting point. The AFPs work by decreasing the amount of ice growth via the Gibbs-Thompson effect. Tahagarabi said this causes the proteins to bind over the surface of seed ice crystals. Additionally, they cause the ice crystals to stabilize and prevent crystallization.
Next, we will be talking a little bit about the evolution and function of these proteins. So scientists believe that antifreeze proteins evolved about 5 to 14 million years ago from a substance called trypsinogen, and this is a substance produced by the pancreas and is converted into trypsin, which is then secreted into the small intestines to break down protein. And originally, scientists came up with this theory because of the similarities between antifreeze glycoproteins and trypsinogen. So some of these similarities are that exon 1 of both the genes are 94% identical. Also, trypsinogen's intron 1 is 2 segments in the antifreeze glycoprotein intron, which show that they have an original similarity of around 93%. And the 3' end of the antifreeze glycoprotein gene is 96% identical to the E6 exon of trypsinogen. Here's just a figure showing the 3 similarities I just stated between the trypsinogen gene and antifreeze glycoprotein gene. Next, I'm just going to talk a little bit about the conversion process that scientists have come up with for the conversion of trypsinogen to the antifreeze glycoprotein. So first, they believe there was a partial gene recruitment of E1, I1, some of E2 and E6 with the 3' UTR from trypsinogen to make the antifreeze glycoprotein, and then everything else was deleted from trypsinogen. Then the main sequence for the antifreeze glycoprotein, the threonine, alanine, alanine, was duplicated to make a tripeptide backbone. And then some of the last things to occur were a 1 nucleotide frame shift and amplification of the backbone to make the antifreeze glycoprotein. And this is just one idea that scientists have. There are a couple that they have come up with that are possibilities for this conversion, but this one being the most likely. Here's another figure showing the three main conversion steps from trypsinogen to the antifreeze glycoprotein, which includes the deletion, recruitment, and amplification to get to that final gene. Lastly, I'm just going to touch on a couple functions or binding techniques that scientists believe may be occurring to keep the fish from freezing in such cold environments. So the first one being that antifreeze glycoproteins are in a left-handed polyprom type 2 helix with the disaccharides all on one side of this helices. And so with that, the hydroxyl groups on the disaccharides are able to form hydrogen bonds with the ice crystals. And when they bind to the ice crystals after adsorbing, the final form changes into a alpha helix. Then the second technique that is proposed is that antifreeze glycoproteins stay in their polyprom type 2 helix, and this forms a hydrophobic and hydrophilic side to the helix. And really, the hydrophobic side is the one that is binding to the ice crystals, specifically the methyl groups and disaccharides on the hydrophobic side are adsorbing to small cavities found on the surface of ice and able to keep those ice crystals from expanding and therefore.
There are two groups of families of notothenoids, Antarctic and non-Antarctic. Each group uses a different type of AFP due to evolutionary differences. The non-Antarctic notothenoids are comprised of three families, Bovichidae, Pseudophretidae, and Eliganopidae. All three of them have evolved in the southern coastal water prior to the isolation of Antarctica. The winter flounder from the Pseudophretidae family uses AFP1s. These proteins have large amounts of alanine, using 23 out of the 24 residues. Additionally, they are often alpha helical proteins. Compared to other AFPs, these are quite large, meaning they have an increased ability to inhibit ice growth. Studies done by Kim et al. Found that AFP1 has a unique ice binding mechanism that is dependent on the size and length of the protein.
On a different note, the Antarctic notothoids are composed of five main groups: the Arnodidroconidae, the Bathrotroniconidae, the Chanotheidae, the Harpagiferae, and the Notodenidae families.
Although each of the Antarctic fish families vary slightly in their diets and behavior, the evolution of their antifreeze protein mechanisms is essentially the same. Many researchers have found evidence that fish's antifreeze protein mechanism stems from a process known as adaptive radiation. This phenomenon describes the mechanism found within each of the families is a result of a specific adaptation to the fish's environment. AFGPs were found within the families between 42 and 22 million years ago. This is likely a result of global temperatures dropping during this time, thus triggering these antifreeze glycoproteins.
Although each family of Antarctic fish utilizes a similar antifreeze glycoprotein mechanism, each group has its own unique evolutionary history. Firstly, the Arnodidroconidae family, or the plunderfishes, is now considered to be monophyletic. In addition, the Harpagiferae family was also conducted to form a sister group with the original family, as shown on the slide. This has been determined and verified by a variety of different studies spanning from 1997 until the present day. However, the specific relationships within these two families has not been explored any further than these preliminary phylogenetic studies. This means that the evolutionary history of these families still remains to be determined. On a similar note, both the Bathrotroniconidae and the Chanotheidae families have an unclear evolutionary history due to poor statistical support from molecular phylogenies. However, it